translation. When a protein is needed by a cell, the genetic code for that protein must be read from the DNA and processed. A two-step process: transcription, synthesis of a single-stranded RNA molecule using the DNA template. One strand of the DNA is transcribed. Translation, the conversion of messenger RNA sequence into the amino acid sequence of the polypeptide. Protein synthesis, both processes occur throughout the cell cycle. Protein, high molecular weight, nitrogen-containing organic compounds, composed of one or more polypeptides. Polypeptides are composed of amino acids. Amino acids contain the following: bonded to a central carbon atom, amino group, NH2, carboxyl group, COOH, hydrogen atom, R group different in each amino acid, typically charged in the cell, NH3 positive, COO negative, negatively charged COO, positively charged NH3. Here we find the amino acid carboxyl group and the amino group. Here the carbon atom, alpha carbon atom, R region different in each amino acids. Structure of the common to all amino acids. 20 different amino acids occur in living cells, abbreviated with 3 to 1 letter codes. 4 chemical group composition of the R group: acidic N, basic N3, neutral and polar hydrophilic N is 6, acidic N2, basic N3. Here the N, the difference in the number of N determine the acidic and basic and neutral amino acids. The neutral and non-polar N is 9, acidic and basic amino acids. Acidic, aspartic acid, for example, glutamic acid; basic, lysine, arginine, histidine; neutral, tryptophan, phenylalanine, glycine, alanine, valine, isoleucine, leucine, methionine, proline. Neutral or polar hydrophilic amino acids: tyrosine, serine, threonine, asparagine, glutamine, cysteine. Amino acids are joined to form unbranched polypeptide by a peptide bond. Peptide bond, covalent bond between the carboxyl group of one amino acid and the amino group of the next amino acid. Here the amino acids: the amino group, carboxyl group, the terminus, C-terminus, polypeptide. Protein shows 4 hierarchical level of structure organization. Primary structure, amino acid sequence, determined by the genetic code of the messenger RNA. Secondary structure, folding and twisting of single polypeptide chain, result of weak H bond and the electrostatic interactions. Tertiary structure, 3-dimensional shape or conformation of the polypeptide chain. Function of the R group contained in the polypeptide. Quaternary structure associated between polypeptide in multi-subunit protein. The example, hemoglobin occur only in 2 or more polypeptide. Here the primary structure, the secondary structure, and the tertiary structure, and quaternary structure. The genetic code: how do nucleotides specify 20 amino acids? 4 different nucleotides: A, G, C, U, T. Possible code: 1 letter, 1 amino acid, 4 amino acid; 2 letter, 4 amino acid; 3 letter, 64 amino acid. The 3-letter code with 64 possibilities for 20 amino acids suggest that the genetic code is degenerate more than one codon specify the same amino acid. Evidence that the genetic code is at replicate code. In 1960, Francis Crick and his colleague studies frame shift mutation in bacteriophage T4 in E. coli induced by the mutagen proflavin. Proflavin adds or deletes base pair, two ways to identify mutant T4 grows with E. coli, the wild type and mutant type, turbid plaque and clear plaque growth with E. coli, and the wild type growth and in mutant no growth. Discovered that frame shift mutation, addition or deletion resulted in different sequence of amino acid. Also discovered that R mutant treated with proflavin could be restored to the wild type relevant. Combination of three mutant routinely yielded revertant unlike other multiple combinations. It's normal single RNA, the amino acid, mutation, single RNA, incorrect amino acid in the polypeptide. How was the genetic code deciphered